Hello friends, myself Dr. Amit Kosti, and today I'm going to talk about the neuromuscular junction. In a previous module I have taught you about the muscle contraction and now how the message from neuron is passed to the muscle that I'm going to teach you, and that comes through the NMJ. So NMJ stands for the neuromuscular junctions. So now from the brains and the muscles a message that you need to contract. I want to move my hand this way, my brain sends the motor neurons, sends the message to the skeletal muscle of my arm that I need to move my hand this way. So from nerves to the muscle, and at what happens at the junction when nerve meets the muscle, that is called the neuromuscular junction. And you would be surprised that this neuromuscular junction is actually a space, a physical space. There's not a direct contact, but there's very minute gap. And to gap in this gap, certain molecules are released that is called neurotransmitter. And that neurotransmitter actually passes the message of contraction to the entire muscles. So let's learn in detail about this topic. So you can also copy, you can also take a snap of these slides, PPT slides. Just a second. Okay. So the neuromuscular junction. Neuromuscular junction. Let me show you the diagram so it will be more clear. You can see here that this is the neuron axon terminal of the somatic motor neuron. So you can see here one of the axon terminal, and these axon, the end point is called synaptic end bulb. So from the one axon at the end where there are many such synaptic end bulbs radiates out. And if you take more detailed view of the synaptic end bulb, what you are going to observe that these synaptic end bulb encompasses many such vesicles. So these are called the synaptic vesicles. Again, I am repeating: axon, neuron have got the cyton. From the cyton, there is axon, and then with the axon terminal, it radiates out many axon terminals, and this axon terminal, sorry, it radiates out many synaptic end bulbs. And in these synaptic end bulbs, what it encloses many fluid, or what is this, many chemical filled vesicles, and this chemical is acetylcholine in this case. So this acetylcholine is the neurotransmitter. So many such neurotransmitter filled vesicles are there in the synaptic end bulb. Clear? And now you see this yellow part is a part of neuron, and what is shown here is the muscle boundary. It's the part of the muscle fiber, it's a part of the muscle boundary, and that plasma membrane of the fiber is called sarcolemma. And this sarcolemma, so this part of the muscle fiber that exposed at the synaptic end bulb is called the motor end plate. Now this motor end plate has got, so this is the actually physical space. There is no touching of the synaptic end bulb with the sarcolemma, but there is a physical gap, a space where these molecules are actually released. So this is the zoom version of this. So what happened at, this is the gap. You can see this area, synaptic gap, there is space. So this is the space, and this is the part of the muscle boundary. So this is called the motor end plate. Now this motor end plate has got the receptor. This violet color, blue color structure is the receptor. So acetylcholine is the ligand, and this blue color structure is the receptor. So it's a lock and key like structure. So when the receptor gets its ligand, there is the conformational changes in the channel. So previously the gate was closed for the sodium, But once the acetylcholine binds to its receptor, what will happen? The sodium molecules present in this cleft pass to its concentration gradient and get passed into the muscle inside the sarcolemma. So when the sarcolemma gets more amount of sodium, it gets positively charged. As it gets positively charged, we say the muscle is excited. So now these excitement of in terms of sodium is passed through entire muscle fiber, and at each part of the muscle fiber through the structure called the transverse tubule get the sodium. So at the interior of the muscle fiber also the sodium moves and carry out the excitation of the entire muscle fiber. And as you know, once the muscle fiber get excited, sarcoplasmic T tubule sends the message to the interior of the muscle fiber, and in response to that, sarcoplasmic reticulum releases its calcium. And once the calcium is released, as you know in my previous module, the muscle contraction take place. So what is the more important feature is that here again I am repeating that the axon has got many synaptic end bulb. Synaptic end bulb possesses many synaptic vesicles, and this synaptic vesicle contains neurotransmitter called the acetylcholine. So when the acetylcholine is released, how does it get released? It released through exocytosis. So once suppose the neuron is itself is excited, its excitement reaches the terminal and this release, and in turn the synaptic end will release acetylcholine in this cleft, and this cleft is called the synapse. In the synapse, when this acetylcholine is released, it will bind to its receptor, and this receptor is what? This receptor is acetylcholine receptor. When the receptor binds with ligand, it gets open and the sodium from the cleft enters into the interior of the muscle, and the muscle fiber, you can say, get polarized, it get excited. Same way as is the propagation of nerve impulse to the neurons. So same like of feature is there, and this is the neuromuscular end plate, and that it get excited. Another feature is that when muscle is actually the muscle end plates, when the nerves is actually getting synapses with the muscle, it is not at the anterior side somewhere to the one end, but in the middle. So what happens when the nerve impulse arises at the muscle get excited in both the direction, forward and backward, because the neurons end plates, muscle end plates are actually located centrally, facing the central. I mean to say the nerves stimulate the muscle at the center. So what happens? The message is passed on the both the side. Excitation gets passed on the both sides of the muscle. So let's study the topic, this topic slide wise. Whatever I will say, I will go through the slide. You can take a snap of the same thing, so it will be helpful for you in exam also. So neuromuscular junction. The neurons that stimulate the skeletal muscle fiber to contract are called somatic motor neuron. What are the somatic motor neuron? Neurons that are under our control. That when we skeletal muscles are muscles that are under our control. If I want to move my hand, I can move my hand, but I don't have such control on my digestive tract muscles. So these muscles, muscles of hand, leg are the skeletal muscle. My tongue muscle are skeletal muscle. So each somatic motor neurons has thread-like axon. So motor neurons are actually remain very in proximity with the muscle. So somatic motor neuron has a thread-like axon that extends from the brain or spinal cord, brain to nerves, or even from spinal cord, these nerves get attached with the muscle. Muscle fiber contract in response to one or more action potential propagating along its sarcolemma and through its system of T tubules. So when the muscle arises at the sarcolemma, it gets propagated and it passes through the interior of the muscle to the T tubules. You know what is T tubule? Transverse tubules that I have already covered in my previous modules. Now the muscle action potential arises at the NMJ, that is neuromuscular junction. Many times in examination they ask what is NMJ stands for. So NMJ stands for the neuromuscular junction. So the synapse between a somatic motor neuron and a skeletal muscle fiber. Where does muscle potential arise? At the NMJ. Muscle potential arise at the NMJ. That is the synapse between the somatic motor neuron and the skeletal muscle fiber. So what is synapse? A synapse is a region where the communication takes place between two neurons or between a neurons and a muscle or between a neuron and a target cell in there. And here in this case, it is the synapse is between the somatic motor neuron and a muscle fiber. Now at the most synapse, a small gap called the synaptic cleft separate the two cells. They are not physically in attachment. Two cells are separated by a synaptic cleft. Because the cells do not physically interact, the action potential cannot jump the gap from one cell to another cell. Instead, the first cell, that is the first cell, communicate with the second cell by releasing a chemical called neurotransmitter. At the NMJ, the end of the motor neurons called the axon terminal divide into cluster of synaptic end bulbs. So axon radiates out many cluster of synaptic end bulbs. Suspended in cytosol within its synaptic end bulbs are hundreds of membrane and cross sac called synaptic vesicles. I have already shown you. Again, let me show you this. So what is there? Just a second. Yeah, here. So this is the synaptic end bulb, and within the synaptic end bulb, there are hundreds of these synaptic vesicles concerning acetylcholine. So that is what is written in the text. So suspended in the cytosol within each synaptic end bulb are hundreds of membrane ankylosing sacs called synaptic vesicles. Inside each synaptic vesicles are thousands of molecules of acetylcholine, and that is the abbreviated ACH. Acetylcholine stands for acetylcholine, that is a neurotransmitter. You might have heard about other neurotransmitters like GABA, dopamine. All these are the neurotransmitters. They have different kind of activity. This is the excitatory neurons that excite, carry out the muscle contraction. So that is not the today's topic. So we will talk about that in some other lecture. So acetylcholine is a neurotransmitter that released in the NMJ. Now the region of the sarcolemma opposite the synaptic end bulb called the motor end plate and is the muscle fiber part of the NMJ. Within each motor end plates are the 30 to 40 million, you can imagine, within each end plate there are not 1, 2, 3, but 30 to 40, not lakh, but million receptors are present. And that is the integral membrane protein. What is receptor? They are integral membrane protein that bind specifically, like lock and key, they bind specifically to the ACH, that is acetylcholine. Now, these receptors are abundant in junctional folds, deep grooves in the motor end plates that provide a large surface area of the ACH. The ACH receptors are ligand-gated ion channel. What are ACH receptors? They are ligand-gated ion channel. A neuromuscular junction thus includes all the synaptic end bulb on one side of the synaptic end plate cleft plus the motor end plate of the muscle fiber on the other side. You can see this structure. These are the motor end plate and these are the structure, and on one side synaptic end cleft and other side these are the receptor of motor end plate. Now, we have gone through this diagram. Yes. Now, a nerve impulse elicits, sense a response, elicits a muscle action potential. How does it sense a message or sense the elicits the muscle action potential? So that mechanism is given here. Number one, release of acetylcholine. Arrival of the nerve impulse at the synaptic end bulbs causes many synaptic vesicles to undergo exocytosis. What is exocytosis? We say cell vomiting. So it carry out the exocytosis. During exocytosis, the synaptic vesicle fuses with the motor neuron's plasma membrane, and liberating the acetylcholine into the gap, into the synaptic cleft. The ACH, that is acetylcholine, can diffuse across the synaptic cleft and that get received by its receptor. And that is the diffuse across the synaptic cleft between the motor neurons and the motor antid. Now the second step, activation of ACH receptor. Binding of two molecules of ACH to the receptor on the motor end plate. So two molecules of ACH get bind to one single receptor, and that reaction causes the ion channel that remain closed to get open for the sodium ion. Once the channel is open, small cation, most importantly, that is sodium, can flow across the membrane. Now, clear? Now, what happens at the third stage? Production of the muscle action potential. The inflow of sodium, and that is down to its electrochemical gradient, make the inside of the muscle fiber more positively charged. These charge change in the membrane potential trigger muscle action potential. Once the neuron sodium is inside, it is charged, it is polarized. These polarize the membrane. These polarize this change in the membrane potential trigger muscle action potential. Each nerve impulse normally elicits one muscle action potential. The muscle action potential then propagates along the sarcolemma and then through the T-tubule system, and this causes the sarcoplasmic reticulum to release its stored calcium ion into the sarcoplasm. And the muscle fiber subsequently contract, because you know when the calcium ion is released, it will bind to the troponin molecule. Troponin gets detached from the tropomyosin. Tropomyosin gets oriented, gets changed. Actin molecules, myosin binding sites get open. Myosin molecules get attached with it. It gets ATP, gets converted to ADP through ATPase enzyme, and there's a release of the phosphate molecule, and then there's a power stroke, then releasing of ADP molecule, detachment to more one ATP molecule and join, detachment, and again the repetition. So that is a part of the sliding filament theory. But for the sliding filament theory to take place, the pre-requirement is this, that sodium get muscle entry get polarized, and that polarized message passed through anti-muscle at the level of T-tubule, and the T-tubule give message to the sarcoplasmic reticulum. Sarcoplasmic reticulum releases the calcium ion, and then the mechanism of the muscle contraction take place. Now, the change in the membrane potential triggers muscle action potential. Each nerve impulse normally elicits one muscle action potential. We have gone through this. Now, the muscle action potential then propagates along the sarcolemma to a T-tubule. This causes the sarcoplasmic reticulum to release its stored calcium into the sarcoplasm, and the muscle fiber subsequently contracts. Now, the fourth point. Now, how does it get stopped? Now, termination. How does termination occur? The termination of ACH activity. The effect of ACH binding lasts only briefly, because ACH is rapidly broken down by an enzyme called acetylcholine esterase. Now, this enzyme actually cleaves the molecule. And where does you find this enzyme? This enzyme is attached to the collagen fiber. Collagen is a protein. So the collagen fibers, acetylcholine esterase is an enzyme that remain attached with the collagen fiber, and this collagen fiber is located in the extracellular matrix of the synaptic cleft. Now, this ACCHE, this acetylcholine esterase, breakdowns ACH into the acetyl part and the choline part. Once it is breakdown into acetyl and choline part, it can no more attach with its receptor, and once it cannot get its receptor, the ion gated channel remain closed, and this way the message is not passed to contract and muscle do not contract. So acetylcholine esterase stop the muscle contraction, stop the excitation of the muscle end plate. If another nerve impulse, but suppose again I want to move my hand, then in case another nerve impulse release more acetylcholine esterase, and as said earlier, and when action potential in the motor neurons cease, that means stop, acetylcholine is no longer released, and acetylcholine esterase break down the ACH rapidly, already present in the synaptic plate, synaptic cleft, and this and the products of muscle action potential, and the calcium releases the channel. Calcium releases channels in the circulatory membrane also get closed. This way, the NMJ is usually near the midpoint of, as I said, NMJ is generally in the near the midpoint of the skeletal fiber. Muscle extension potential that arise at the NMJ propagate toward the both direction of the fiber, and this arrangement permits nearly simultaneous activation, and that means the contraction of the all muscle fiber at once. Now, this is something for your understanding. You have understood that many film stars get their what you call their skin tone, some Botox treatment you have heard about. So let's talk about that. How these sometimes toxin helpful. Several plant products like drugs selectively block certain events at the NMJ. When the neuromuscular junction is blocked, what will happen? Botulinum toxin, there's a toxin, a poison, a toxin. Botulinum, produced by the bacterium Clostridium botulinum, block the exocytosis of the synaptic vesicle at NMJ. So you can understand, you can imagine the neurotransmitter doesn't get released at the NMJ. So what will happen? No muscle contraction will take place. So it blocks the exocytosis of the vesicle at the NMJ. As a result, ACH is not released and the muscle contraction does not occur. And you know, certain muscles are actually required to get contract. The bacterium proliferate, it improperly cannot. Where do we find this bacteria? It is found in improperly canned foods, and the toxin is one of the most lethal chemical known. A tiny amount of this botulinum can cause the death by paralyzing of the skeletal muscle. Breathing stops. A person will not be able to breathe. Stop due to paralysis of the respiratory muscles, including diaphragm. When diaphragm muscles get paralyzed, the person cannot breathe. And if there is no breathing, there is a death. Yet, it also the first bacterial toxin to be used as a medicine. Botox is the name of the company. The injection of the Botox into the affected muscle can help the patient who have crossed the eye, uncontrolled blinking, or spasm of the vocal cord. So the treatment of these things, this toxin is used, and that interfere with the speech, vocal cord treatment. It also used as a cosmetic treatment to relax the muscle that cause the facial wrinkles. So many film stars, they remove their wrinkles. So they get the treatment from the botulinum injection in very minute quantity, and to alleviate chronic back pain due to muscle spasm in the lumbar region. So the toxin is also medicine. Now this is the briefing of the what entire take place. You can see here the nerve impulse. Then this is the end plate. Muscle end plate. This is the white color. This is synapse. This is the pink color structure is acetylcholine. This acetylcholine is released in the cleft. It is welcomed by its receptor when two molecules is of the acetylcholine bind with its receptor. Sodium molecules get inside the entry. Once the sodium is inside, the muscle get polarized. Its message is passed to the entire muscle. At the triad, you can see this is the triad T-tubule sarcoplasmic reticulum. This is the part of terminal cisterna. It will release to its channel the calcium molecules. Calcium molecule will bind to the troponin molecule. Troponin will get dislodged. It will make the conformational changes in the tropomyosin. Now, tropomyosin will no more hide the myosin binding site on the actin molecule. As the myosin binding site on the actin molecule are exposed, myosin will get attached. At the same time, ATP will bind to the myosin molecule. ATP get converted into ADP and phosphate, and then the few steps of the muscle contraction, and then the power stroke, and then this will happen. So this is the muscle, and then once the signal stop, then there is no more calcium ion, and the muscle contraction stop. Then there is a relaxation of the muscle. So this way, muscle contraction take place through the NMJ. So neuromuscular junction, NMJ we talked about today in this module. To neuromuscular junction, again I am repeating, this is the neuron and axon, and this has a axon terminal have got the synaptic end bulb. This synaptic envelope has got the synaptic vesicle. It has got acetylcholine. This acetylcholine release into this physical gap that is called synapse. This part is called the motor end plate. Motor end plate got plasma membrane, and this plasma membrane has got millions of this receptor. Two molecules attached with the receptor. Once the receptor attached with the moist ligand, it gets open, more and more sodium gets inside, sodium gets inside, muscle is polarized, calcium ion gets released from the sarcoplasmic reticulum, and you have muscle contraction. So this is all about the synapse. I hope you have enjoyed, you have learned this lecture. You can take a snap of these slides. Okay, you can draw simple diagram. You don't need to, you are not, this diagram is not required in examination. Okay, I hope you understood the topic. So here I'm ending this lecture. Good luck for your exam.